Hey everyone, and welcome to Microbial Minutes. This is the American Society for Microbiology's every couple week update on what's hot in the microbial sciences. I'm Julie Wolf with the American Society for Microbiology. These are the Microbial Minutes for October 16th, 2018. Today, we're going to talk about a bunch of different stories that have been resonating in both the news and research communities. The first is going to be on viruses and their role in addiction. And the next story will be also about viruses, but a very ancient virus. We'll then move underground to talk about some underground cyanobacteria. And then we will move to the honeybee microbiome, finishing with some updates that have been in the news recently on vaccines. So let's dive right in. Our first story is from the Proceedings for the National Academy of Sciences, or the PNAS journal, and it's titled Human Endogenous Retrovirus K HML2 Integration with RAS GRF2 is associated with intravenous drug abuse and modulates transcription in a cell line model, which is a mouthful, but does basically say what their findings are. To summarize more succinctly, the too long didn't read, the title summary is that retrovirus found within gene may influence addiction. So, what is a retrovirus? Retroviruses are viruses that are able to move from the RNA genome form and integrate as DNA into their host genome. I have a schematic on the right hand side showing retroviral, at this case DNA, which is going to integrate sometimes somewhat randomly. Sometimes it only requires a particular base, like maybe a two base recognition site. And sometimes it has a very specific requirement, really depends. And we're talking about a retrovirus called the HERV HML 2, or as the authors call it in their paper, HK2. HK2 is an ancient retrovirus remnant, which means that it's not actively able to make all of the viral proteins and parts necessary to make progeny viruses, but that this DNA, the viral DNA, remains as part of the human genome, but in various sites in different people. So not all people have the same integration sites for these HK2 viruses. Now, one uncommon HK2 is found occasionally inside of a gene called RAS-GRF2, which is involved in dopaminergic activity, or dopamine transition. Dopamine recognition. And dopamine is a neurotransmitter through which neurons can communicate. So it's important for brain activity, among other things. And this particular gene, RAS-GRF2, has been identified through a gene-wide association study. So looking at characteristics of a large population and trying to identify a particular characteristic that may be associated with a particular genetic sequence. A SNP in this RAS-GRF2 has been associated with alcohol addiction. So there's already an indication that this gene may play some role in addiction. But that SNP, that single nucleotide polymorphism, is located between exons 3 and 4. So the exons are the parts of the genes that are actually put into the eventual protein product. And that is in a different part than where this HK2 is integrated. HK2 is integrated in between exons 17 and 18. So this is a very large gene. It has a lot of different sequences that are going to become part of the final mature mRNA that are eventually incorporated into the protein product. And this integration site is not in the same place as that SNP associated with alcohol addiction. However, because the alcohol addiction has been associated with that particular aberrant sequence, the authors wanted to ask the question of whether this HK2 integration in this particular site is associated with addiction of some sort. And so they did this by looking at several different population sets. They wanted to screen using PCR to look at whether there was an integration. As you can see on the right-hand side, this is the schematic for their screen. They're looking at whether individuals have a full length form. So they have two sets of primers. Really, it's three primers that will differentially amplify the sequences that people have. So in every individual, They're looking for the full length form, which is going to be this forward and this reverse, and is going to give the larger band, as you see here, in every individual. So these different numbers are different patients whose sequences they're interrogating. And then if there is an integration, they have one primer pair that's specific only for this HK2 retroviral insertion. And so this smaller second band will only appear in the case that there's been an integration of that retrovirus. And you can see that here and here. So two of the patients that they're looking at here have that smaller integration. Now, remember, everybody has two different copies of their alleles or of their genes. They have one allele from the mother and one from the father. And so, even in these patients that have a retroviral insertion site, they also have a larger allele as well, suggesting that they are heterozygous, meaning that they have one integrated site and one non integrated site. And so in this, what the researchers used this scheme for was to screen two independent populations of people who inject with drugs. So these are PWID, as they're referred to in the text, and there are two different populations that were independently interrogated. One is an HIV infected population from Greece, and one is an hepatitis C infected population from the UK, from Scotland. And these different populations were interrogated to make sure that there was no effect of perhaps a similar genetic background that could be found within those different populations. And both within the HIV infected individuals and with the HCV infected individuals, there are some who are more likely to be persons who inject with drugs. And then, of course, there are those that are not injecting with drugs. And what they found is that the people who do inject with drugs within these two independent um, populations, they are 2 to 3 times more likely to have this particular integration. So to have the retroviral integration that would give the two bands instead of the single band. Now, this is a pretty rare integration in general. The general population has between 5 and 15%. I believe in their control populations, they saw about 4% of the individuals who had this integration. So that does mean that there's still a lot of people who are injecting with drugs that do not have this integration, this particular retrovirus. So, what could this retrovirus be doing? The authors then wanted to look at whether there was an effect of having this integration on the RAS GRF2 transcription. And so they used CRISPR in order to insert this HK2 into the exact same site within a cell line model, so not within any type of patient or animal model. And then they looked at the transcription of that RAS GRF2, and they saw that as soon as that retrovirus was inserted, that they did see a decrease in transcription. So that could potentially affect the transmission of this or the reception of this particular neurotransmitter, dopamine, as it's being secreted between neurons. However, they saw that the transcription gradually increased the longer that HK2 was, um, the longer the recovery time was after that insertion had been made, indicating that this might not be the entire story. Now, when they sequenced the HK2, they did see a few patient variants that were what they called coding competent, meaning that they could potentially make progeny virions and uh, infect other cells and perhaps even jump into a new place within the genome. And so, from this, the author said that HK2 might even be considered conditionally pathogenic, of course, depending on whether or not that variant that one has is able to jump from or to make variants, to make progeny viruses and jump into a new place within the genome. So this was picked up in a whole number of different mainstream news outlets. CNN, The Economist. It was shared by a number of prominent virologists on social media. The primary author, Major Kinis, said that I believe a significant part of this unexplained disease diversity will be will be understood by studying this difficult to understand dark genome. And this dark genome is referred to several times in these news outlets, and that is the ability of different types of viral remnants, not necessarily active retroviruses, but some of the transposons or retroviruses that have integrated into the human genome to differentially regulate genes that are part of that are found, the coding sequence is found within all individuals, but perhaps have differential transcription based on where those insertions have been found. I also liked the wording in The Economist because, of course, making these types of correlations is a strong argument, but it's not causative. And The Economist summarized this nicely by saying that these observations and experiments do not absolutely prove that HK2 plays a role in addiction, but they make it likely. And of course, prove is a word that many scientists do not like to use. They often prefer the language of this supports the probability of HK2 playing a role in addiction, not necessarily proves anything, since a good scientific hypothesis can always be disproven. All right, moving on, we're going to talk about another ancient virus, but this is one that was found within some ancient samples. This was a study from the American Society for Microbiology's Journal of Virology titled A 1,000-Year-Old RNA Virus. So, pretty much says what it is, but the perhaps more detailed summary would be that the oldest known plant virus was found in ancient maize cobs. So, one of the shocking things or surprising things about this particular virus that was found is that it is an RNA virus. RNA is much more labile than DNA. There are lots of DNA studies done on ancient specimens because DNA is a very stable molecule that can withstand uh, a lot of different conditions and maintain its integrity, whereas RNA is much more easily degraded. For this reason, there are a few RNA viruses that have been discovered in archaeological samples. Most of the viruses, the ancient viruses that have been found, are DNA viruses. In this case, the authors looked at ancient maize cobs from an archaeological dig site in Arizona. They investigated 312 different maize cobs. And from these maize cobs, they took a small specimen and enriched for double stranded DNA. And then this double stranded DNA was converted to DNA. Now, they got the age of the maize cobs by using radiocarbon dating and estimated that these maize cobs were somewhere around 1,000 years old. From these double stranded RNA samples converted into DNA, they were able to identify a novel plant virus with a double stranded RNA genome. And this double strandedness may have helped the RNA to withstand the test of time a little bit more than some of the single stranded RNA viral genomes that would be more easily degraded. Now, three of these samples had a nearly complete viral genome. And from this sequence, they were able to name the virus Zea mays chrysovirus 1, or ZMCV1, which is now the oldest known plant virus. So, this chrysovirus is a known family of viruses. It is a persistent plant virus that is often transmitted through the seeds. So, this isn't something that is passed between different adult plant species or plant members, but is passed from the parent plants to their progeny through their seeds. And so, it's vertically transmitted rather than horizontally transmitted. Now, this chrysovirus is something that chrysoviruses are still found today. And when looking at modern corn species, the authors found that there was a very closely related chrysovirus that was only 3% divergent from that which they found in the ancient samples. And you can see these highlighted in this phylogenetic tree here, where the ancient ZMCV1 viruses are only 3% divergent from the current modern corn chrysoviruses highlighted in black here. This was surprising to the authors because it undermines the classic view of RNA viruses, which are thought to replicate with low fidelity and thus diverge quite quickly. However, there has been very little divergence in these samples, the ancient and the modern samples here. However, there was no ZMCV1 that they found in teosinte. And teosinte is the precursor organism from which corn is thought to have been modernized and turned into the crop that we know today. And so there are a couple different explanations for this. They looked using both PCR amplification, using degenerate primers, as well as northern blot to look for similar sequences within the DNA themselves. So you can see here in maize and here in teosinte, there's a signal in the maize, but not in the teosinte. And this is possibly because the maize ancestor, after it split from teosinte, is what was infected. And so those maize cobs, the ancient maize cobs, were infected, and that was passed down into what we now know as modern corn. Or it's possible that the sequences have diverged too far to identify by these molecular means, where the probes just won't hybridize with the teosinte genome. So this was not a widespread news article, but it was shared by a number of virologists in the social media scene. And so I thought that this was worthy of discussing because of its novelty of the double-stranded RNA genome, as well as the close relation between the ancient and the modern double-stranded RNA virus genomes, between the maize cob virus and the modern corn virus. Moving on, our next study is again from PNAS, and this one is titled Viable Cyanobacteria in the Deep Continental Subsurface. And so the take home message is pretty much as the title states: cyanobacteria can be found in deep subsurface. And this is pretty surprising, not only because cyanobacteria are known to be quite common in the surface area, but also because they are well known to be photosynthesizers. They make a lot of our oxygen that we breathe. And so, what this group wanted to ask was, what about where the sun doesn't shine? Are there cyanobacteria? To be honest, they weren't really looking for cyanobacteria specifically, but were interested in what types of microbes might be able to live deep below the surface area where we walk. Now, caves have already been one niche in which cyanobacteria have been found. However, caves, even in a cave system, some people think that low levels of photons, which are found in those niches, are still able to allow the cyanobacteria to undergo photosynthesis. However, what these authors here were looking at was not caves, but the subsurface rock below our feet. And so what they did was to drill rock core samples at 420 and 607 meters below the surface. These had to be taken extremely aseptically because, of course, it would be potential to contaminate these deep core samples with the surface soil. And soil is really not something that is found at more than a meter or so on the surface of the earth, but contains a large variety of different microbes, which could potentially contaminate those core samples if the drilling is not done very carefully, as well as, of course, all of the processes to purify DNA. All of these had to be done under extreme aseptic techniques to make sure they were truly sampling the rock core samples and not any of the microbes which might inhabit um, the air or the equipment which was being used to purify them. And so after they purified the DNA from these rock core samples, they looked at the microbial profiles. And they did this by 16S ribosomal gene sequencing. And you can see here at 420 and 607 meters, they had quite similar profiles where the largest proportion was, surprisingly, these cyanobacteria. They also found other microbial groups, such as the Ascomycota fungi. They also found other bacteria, such as the Bacteroidetes and the alpha proteobacteria. In addition to looking at the types of microbes by gross characterizations, they then looked at the cyanobacteria clades themselves to look more closely at what types of cyanobacteria might be found in these conditions. And so they're showing here at different depths. So on the y-axis here, we're looking at depths, and then we're looking at the proportion of that sample which was found to be a particular cyanobacterial clade. And interestingly, they found that these cyanobacteria were most prominent in areas where there was very low hydrogen. So they were at the same time looking at the chemical composition of these particular microbes, and they found that the cyanobacteria were most common. In fact, all the microbes were most common, not within the rock, but along tiny little fractures that ran deep into the surface of the earth. And along those little fissures, they were able to look at the chemical composition, and they saw that the hydrogen was decreased in the regions where the cyanobacteria were most prominent. And we'll get back to this in the next slide. So they looked at, in addition to the 16S ribosomal gene sequencing, they looked at the entire genetic sequence of what was there by metagenomics, and they were able to identify a number of different genes important for cyanobacterial metabolism, for things that are important for nutrient acquisition, such as nitrogen fixation, for processes such as respiration, and looking at what their final electron acceptors may be in this extreme chemical environment, looking at potential for iron or nitrous oxide to be an electron acceptor during respiration. And they also found the genes that they identified as proposed hydrogenases. And so the hydrogenases of these cyanobacteria may allow them to use hydrogen as an energy source. And the researchers didn't show directly that these bacteria are able to um, use hydrogen as an energy source. But the fact that they had these genes, combined with the fact that the cyanobacterial populations were highest, where the hydrogen concentration was the lowest, as I showed you in the last slide, had them propose that this may be how they are getting their energy in the absence of light, because although there are fissures going down into the rock, there is not light. There's no sunshine that may allow them to undergo photosynthesis. Although some of the isolates did have the genes that would be necessary for photosynthesis should they all of a sudden become exposed to the surface. And so this was picked up by a number of science related outlets. This was picked up by Science News, where the study co author, Fernando Puente Sanchez, said that the team discovered the cyanobacteria, but that this discovery was unexpected and counterintuitive because these scientists had, like many scientists, expected cyanobacteria to be most enriched where there was sunshine with sunlight and they could undergo photosynthesis. And this was also highlighted as a potential tie to life in extreme environments such as Mars. So the region in which these scientists were digging or doing their rock core sediments was in the Tinto region, which is thought to be similar to Mars due to its abundant iron and sulfur materials. Now, of course, Puente Sanchez was quick to say, I'm not claiming that there are cyanobacteria on Mars, but this does play an interesting role in thinking about the possibility of where life may exist in extreme environments, such as on Mars, or what might be possible should Mars be colonized and seeded with bacteria from Earth. All right, we're going to move on to our next study, also from PNAS. They're just on a roll the last couple of weeks. And there is a study in PNAS, and then there was one shortly after in MBio on the same subject. These are both from the same lab, looking at honeybee um, interactions with glyphosate. So the PNAS study is titled Glyphosate Perturbs the Gut Microbiota of Honeybees. And the bio study is titled Pathogenicity of Serratia marcescens Strains in Honeybees. And so the take home message of these two combined studies is that glyphosate alters honeybee microbiomes and increases their susceptibility to infection. So we've talked on previous microbial minutes about how honeybees are dying. This is an extremely serious problem. They are dying at such a precipitous rate that a relief act was brought by honeybee farmers before Congress in 2017 in order to address what is known as colony collapse disorder, where colonies will undergo an extreme population decrease in a matter of days or weeks, not just disappearing, but actually dying. And the cause has been pinpointed for. There have been a number of different causes which have been proposed. Now, a lot of these may be acting together, and we'll talk about that at the end. But what this study is testing is glyphosate, which is a common weed killer used largely by Monsanto. It's famous as Roundup, but of course, other individuals and companies use this as well. Now, the target of this molecule, glyphosate, is an enzymatic process which is found primarily in plants. That's why Monsanto and others are able to use this as a weed killer. It will kill all plants that are not genetically modified to be resistant, and thus whatever crop one has sown within the fields is able to resist that particular mode of action. But some microbes also have this same enzymatic process, particularly those that are the symbionts of honeybees and are found in the honeybee gut. And so the first question that was asked in the PNAS paper is whether exposure of honeybees to glyphosate can alter their gut microbiome. And the answer was yes. So, here looking at individual bees along the y axis, the researchers looked at both the relative abundance and the absolute abundance. So, this is a very nice way to look at a gut microbiome because first it looks at the total number of microbes. And you can see that most of the honeybees, which were treated with glyphosate, as you can see here in this shaded box, have a lower abundance in general, meaning Um, lower CFUs, lower numbers of bacterial cells within their gut, with of course a few outliers here. But if we normalize all of these to 100%, we can look at the proportions of the different populations that make up whatever that absolute number is. And here's where it becomes more apparent that they are lacking some key players, particularly this Snodgrassella is pointed out as being lacking or decreasing in these glyphosate treated bees, and that these glyphosate treated bees were also more susceptible to Serratia. Serratia is a natural microbe that lives in the environment and at very low numbers can colonize the honeybee gut microbiome. However, in part because of the decreased abundance and also because of the change um, in the alteration of the composition, the honeybees that were treated with glyphosate were more susceptible to the serratia, as you can see here, than those that were not treated with glyphosate, which can still succumb given a high enough inoculum, but are not nearly as susceptible as those that have had their microbiomes altered. What I'm not showing you is the data from the MBio paper, which looked at different species of Serratia and found that it was specifically Serratia marcescens, which was the primary pathogen. It's an opportunistic pathogen, but was the main opportunistic pathogen, which was taking advantage of the altered honeybee gut microbiome state, and that the honeybee immune system does not seem to recognize and fight off Serratia marcescens very effectively. And so part of the innate immunity of the honeybees is to have a particular composition within the microbiome, which is able to keep those numbers, the numbers of Serratia bacteria, very low until they are treated with glyphosate, and in which case those that are um, colonized with Serratia marcescens are likely to succumb to that infection. And so this was picked up in many, many different outlets. Uh, the It was picked up in places such as Newsweek and The Guardian, and even rags such as the Daily Mail, which we've highlighted before. It was a good lesson in science communication. You can see the title of the Newsweek article as well as the caption of the image on the Daily Mail. Both state that the weed killer glyphosate is killing honeybees, which makes it sound like the molecule itself is killing the honeybees, whereas it is really increasing potential stress or making them more susceptible to an infection that they would otherwise be able to ward off. That was nicely conveyed by the lead author, Mata, who stated that understanding how environmental stressors affect honeybee health may be crucial to reverse or control the high mortality rate of bees observed in colonies in many places in the last decade. And this data, which is released in PNAS and MBio, also complements some studies which we didn't cover in Microbial Minutes earlier this year, but that suggested that glyphosate also influences honeybee larva development and may impede and lead to larval death. And so that is something that may be additive with a number of other potential sources of environmental stress for these honeybees, and something that we will be watching in future Microbial Minutes. All right, our next journal article comes from the Canadian Medical Journal and is titled Population Level Sexual Behaviors in Adolescent Girls Before and After Introduction of the Human Papillomavirus Vaccine from 2003 to 2013. Take home message from this study is that the HPV vaccine doesn't affect teen sexual behavior. So, we're highlighting this because HPV vaccination rates are something that we've discussed previously on Microbial Minutes. The rates are increasing. They've increased about 5% in the last couple of years, so that now nearly half of 13 to 17-year-olds have received their full vaccination, and almost two-thirds have received the first dose, which is great. However, the HPV related cancers are also increasing. So, compared to 30,000 cases in 1999, in 2015, there were over 43,000 people who developed cancers that were associated with HPV infection. And so, the ability to ward off cancer is something that we can do now, given the HPV vaccine. It can prevent cancers in both men and women. And these are often cancers that are associated with an infection that they may have acquired much earlier in their life. And so this is a sexually transmitted virus and can lead to things such as ovarian or cervical cancer. But the idea that one would give a vaccine to young teens has been met by opponents who state that this will encourage teens, the vaccination will encourage teens to have promiscuous sex. And so this group con- conducted a survey of 300,000 girls, only asking the girls about their sexual behavior in 2003, 2008, and 2013. So 2003 is before the vaccine was released. 2008 is just after or just around the time that the vaccine was being released, but not very many people were, not very many young people were receiving it. And then 2013, about 5 years ago, there were not quite the numbers that I just quoted, but it was more common for young people to have this vaccine. And so the sexual behavior questions were asked, and the proportion saying yes is stated here. You can see that the numbers have stayed pretty even across the board. So, whether they have ever had sexual intercourse, from 21 to 20 to 18%, with some variation in probability here. Whether or not they used condoms at their last intercourse, whether or not they were on birth control, these numbers, whether they'd ever been pregnant, in fact, seems to have gone down just a little bit. But more or less, the conclusion from this is that the availability of the HPV vaccine has not affected what has not affected the, teen sexual behavior. And so this was picked up and published just yesterday. It was published in USA Today, where they quoted the lead author Ogilvy telling Newsweek that when parents ask why they are hesitant to have their children receive the vaccine, one of the key issues they identify is the concern that the vaccine will encourage children and adolescents to make poorer sexual health choices. And a number of different experts in this article are cited as saying that teens may actually be making better sexual health choices due to the information that they receive. And so this was found in a number. USA Today is about as mainstream as you can get in the United States. This is also shared widely on social media. I liked the way that this particular healthcare provider and medical practitioner stated it, saying that vaccination does not encourage promiscuous sexual activity any more than fire extinguishers encourage people to set fires or seatbelts encourage people to have car accidents. A little bit hyperbolic, perhaps, but stating that the behavior is not necessarily determined by the protection. This also correlates or not correlates. In related news, there was a new FDA approval for the HPV vaccine. Now, before, what it had been approved through was young people through aged 27. And now that has been expanded from 27 through 45 years old. And that's important for insurance cost coverage. So, although one may not have been exposed to HPV before 27, it would be very hard to get the insurance companies to pay for the vaccination if the FDA has only approved it through age 27. So this will allow people to perhaps be protected. And I think studies will be able to determine whether or not vaccination can protect people who may already be infected. So one final study, not study, one final story, which is a very sad and unfortunate one, but something that I wanted to highlight since it broke late yesterday, is that we have entered the 2018-2019 influenza season, and we've now experienced in the United States the first death, the first pediatric death due to influenza. This is in a child in Florida who was unfortunately not vaccinated. And I'm raising this point because it is time to get vaccinated. We talked about this just last time, that the time for maximal protection through the worst of flu season is now late October, to protect you through the peak, which is going to occur in January and February. Now, even if this child had received their vaccination, it's possible they wouldn't have been protected, because it does take a good 2 to 3 weeks for the immune system to ramp up and create that cellular memory in order to recognize the invading pathogen. But it's very important to get the vaccine, since last year, 80,000 Americans died from influenza infection. And that was a particularly virulent influenza year. Indications from the Southern Hemisphere suggest that it will not be quite as bad or as severe an influenza season this year. However, there are always people who are not able to become vaccinated due to their immunocompromised states. And one of the reasons that people with healthy immune systems get vaccinated is to protect those who are more vulnerable. All right, so that's going to wrap up our microbial minutes. If you liked it and you want to watch it next time, make sure to subscribe there at the bottom. To recap what we talked about, hold on, there seems to be a breaking question, which is, are there indicators that glyphosate makes humans more susceptible to infections? There are no studies tying human infection and glyphosate. Glyphosate has been in the news. You may have been following that there are some people who think that it is a carcinogen. There was just a farmer who won a case against Monsanto for being exposed to glyphosate and developing, I believe it was a blood cancer, although I don't recall off the top of my head. That case is still being litigated. But it is, as I mentioned, listed by the World Health Organization as a potential carcinogen, as is, I should mention, red meat. So there are, the doses in the poison, or the poisons in the dose, excuse me. All right. So to wrap up what we've talked about on this session of microbial sciences, we first discussed how a retrovirus found within a gene, the RAS-GRF gene, may influence addiction or predilection to addiction. We talked about the oldest known plant virus, which has been discovered in an ancient maize cob. We discussed the new story that cyanobacteria abound in the deep subsurface. We talked about how glyphosate alters a honeybee microbiome and increases susceptibility to infection. And we wrapped up with a story about how the HPV vaccine doesn't affect teen sexual behavior or practices. I'd like to thank everyone for listening, and thank you for the question. If there's anything else, feel free, if you're watching this not during the live session, to leave a note below, and we will get to answering your comments or questions later in the week. And I'll talk to you next time on Microbial Minutes.